I photographed the back of the T-shirt of one of the staff, and this title is still puzzling me. I don't understand what it means. It's very vague. For me, it has something to do with the communication, the way we talk to each other. Communication for life. We communicate to live. We live to communicate. We leave the world to make an impact on people around you. In fact, I'm going to do a simple practice now. I want to make sure that you're going to listen to me attentively for the next 17 minutes. I want you to pull out your cell phone and turn it on. Yes, turn it on. And I want you to call this number and to see what happens. Let's see if anyone can tell me what happens. All right, go ahead. Hello? Hello? Who is this? Hi. I'd like you to tell me what the size of your waist. Okay? 25 inches. Okay, that's good enough. Yeah. All right. Now, I want you to imagine that I take your waistband and add 1 foot to it. So it's about 12 inches longer. And then let it loose. So it's going to be hung like this, right? It's going to be uniformly loose around your waist. And the gap between your waist and the waistband will be about, you can work this out. It's about 2 inches. You can sort of imagine 2 inches all around your waist. Okay, thank you very much. Listen more to what happens. Now, thank you very much. You can turn it off. Now, what I want you to imagine is, let's do the same practice to an elephant waist. A couple of, about maybe 10 feet around the waist. Let's do this actually to the Earth. Earth has a waistline of about 26,000 miles. We are going to do the same practice. We're going to take the waistband and then add 1 foot, 12 inches to it, and let it loose. So let it loose all around the waist, around the equator, equally around it. How much gap do you think you're going to have between the surface of the Earth and the waistband? Anyone knows the answer to this? Most of you think it's insignificant, so thin, right? Does anyone disagree with this? Anyone? Yeah, tell me the number. Okay, the gap between the waistband and the Earth is 2 inches. That's the power of one. The one who knows the answer will give you the inroad to the new realization, and most of us, including myself, is so biased that we are very good with the intuition. Your intuition for this waistband was incorrect. Now I'm going to wake you up, really. Scientists are having fun. One of my students came back from Las Vegas with this photograph. I'm not going to tell you for the privacy of her privacy. Now, if the communication goes wrong, you get one of this bulky muscle. This is a real photograph from Belgian blue that has a problem with one of the communication language that goes on in the body. It's called GDF8. It's a gene, but you don't have to know the name exactly. It's one of the sentences that tells the body to bulk up your muscle, lose the fat, things like that. And that's what I'm going to tell you about, how we communicate. We all know we start out with a single cell. We all start out as an embryo. What's my idea about these new communication tools? We want to improve our communication in the body to have a better life. And that's what I'm going to tell you about. We all start out with a single cell. During the developmental process, about 9 months in the mother, we develop tissues, and these tissues talk to each other. And that tissue get together, and we become a body, and this body is designed to make a living. But what I want you to understand and recognize is that this gene, the gene we all carry, have a code to terminate this life 100 years later. It's true. It's sad, but it's true. It's part of the program. It's not an accident. We are designed to die after about 100 years. So clinically, though, during this communication process that you see between the body, from one cell to another, from brain to your pancreas or things like that, when that communication breaks down, and that's the natural one-way street to terminate 100 years later, and once in a while we have a trouble, clinical trouble. Our heart is not working very well, or our pancreas is not working very well. We develop diabetes. So we want to have a second chance. So in the past 20 years or so, the new technology called stem cell technology is giving us a second chance. We have a chance to time back to an embryonic state in a petri dish, not an embryo anymore. In a petri dish, we start out, and then we have a second chance to recreate the new tissue and possibly new body, and then we can improve our system. What's missing, though, is the communication language. Those process noted in red arrow is not really a natural process. There's no language to tell the cell to go back the time and restart the game. It's not part of our gene. So we need new language, synthetic signal, new language that can tell the cell to go back and restart. That's what I wanted to do, and that's what I'm going to tell you about. Just as an example, I'm going to tell you about just one sentence that we created. It's called AB204. How do they talk to each other? I mean, they don't really talk through a cell phone. They don't really do a texting to each other. They talk by binding together. So this is the kind of system. This is the situation that you're going to run into. If you are one of those sentences that goes from one cell to another, you're flying in, and then at the destination, you bind to a molecule called receptors. It's shown in green and purple. Those are the receptors that catches you and recognize through the codes encoded on the surface of this molecule. And that's what we do in the lab. I don't talk about that anymore, but I don't talk about that today, but I will just tell you about those languages shown on the right side, and that's the molecule that I'm going to talk about. It's like a butterfly. What do we want to do to create a new sentence? It's like creating a new sentence is like creating a new molecule. Here's how we do. I'm showing two lines here, two sentences. It's a long amino acid sequences, but consider that it's just a sentence to tell the cell to change to a bone or things like that. There are two lines, and I'm switching those words. I'm underlining them. And that language is going to be the new sentence that I call AB204. So in other words, AB204 is made up half with the B sentence and half with A sentence in the order of B-A-B-B-A-A. Baba, baba, right? B-A-B-B-A-A. It's like making a new potato man using A and B by changing its limbs and arms and legs, creating C. And that C sentence is going to do something else that we hope is useful for us. If you didn't understand what I just said, it's like creating a new butterfly. Here's butterfly that you find in nature. It's many different shapes and colors, and you're creating a new chimeric butterfly, and hopefully this new chimeric butterfly will do something that you want. Here's one data that you find in the cell culture. If you look at the top line, this is what happens to those cells ready to go over a 1 week time period. If you look at the second row, that's what happens from left to right. When the cells turn into a bone, they become darker, and it's stained in black, and you can see sort of staining going on in 1 week time period. If you look at the bottom row from left to right, you can see what happens if that signal happens to be AB204. You can see a lot more bone is going on. Is that really true? We drilled a little hole on the top of the skull of a mouse, and then if you don't do anything over a 3-month time period, that drilled hole stays the same. That's the left picture, and you can sort of see the X-ray picture right underneath. And nothing happened. With the natural sentence called BMP2, that's a bone morphogenetic protein, it creates the bone. With the natural signal, you can see some recovery after 3 months. You see an X-ray picture in there too. If you look at the right side, that's with the new signal that we created, AB204. You can see X-ray picture of that. You have a very formidable recovery of the bone. You do that with the mouse. Now this is a tibia bone. You make a little fracture, and that fracture is going to be The same in your left picture. It's what nothing happens if you don't do anything. You can see a little gap in the left leg. And if you look at the picture in the middle, that's what happens with a natural sentence, BMP2. You have some partial recovery. If you look at the right picture, then you can see AB204 is very powerful in creating a repair, basically a bone fusion. We want to use this for spinal fusion. Now I'm going to tell you what we are going to use it for. This is a rabbit spine, and when we have a spinal problem with our spine, we have a lower back pain, and that's because our disc is worn out between vertebrae number 4, number 5. We have 33 vertebrae bones, and this is how you can do the test with the rabbit, in this case, rabbit fusion. So when you have a pain, that disc is worn out, and then that touches the nerve, and that causes the pain. And by removing that disc and fusing those bones up and down, you can actually take care of that back pain. Actually, this is what people do currently. Listen to this.
The spine contains 33 bones or vertebrae, which support and protect the upper body and spinal cord. Injury to the spine, such as a herniated disc, an abnormal curvature of the spine, or an unstable spine caused by illness, may require spinal or vertebral fusion in order to reduce pain and improve mobility. One method for vertebral fusion involves implanting small pieces of the hip bone between the injured vertebrae. These small pieces of bone will combine or fuse with the existing vertebrae to create one solid bone. Until the bones fuse together, the spinal column must be kept immobile. Oftentimes, a metal rod is inserted to keep the spine in place. An external cast or brace can also be used for additional support. After vertebral
So that's what they do in actual case. And we think we can do this better by not having a first surgery to cut the hip bone out of hip. And you go straight to the area where you want to do a surgery. And then by using a piece of sponge soaked with your protein called AB204, you can have a better, stronger spinal fusion. That's what we currently pursue. Now, that's one example. That's just a way of creating a new synthetic molecule. Synthetic molecule is a way to communicate between different body parts from one to another. I just gave you an example with the bone. We're thinking of doing something else for other parts of the body. I don't have time to go over that. Ultimately, what we like to do, so I gave you the example of AB204. Ultimately, what we would like to do is to open up our communication space to other life forms in our body. Those are the bacteria in our guts. In our guts, we have 10 times more number of cells than our body, and those are bacteria. And Bacteroides is one of the most successful symbiotic bacteria that lives in our gut from day 30 from the time we were born until we die. And we're trying to do engineering of that Bacteroides to do something good for us. I don't think I have time for that. And ultimately, the imagination takes me to synthetic animals. And this Cheshire cat is a sort of a symbol of my synthetic animal. In my lifetime, I'm hoping to achieve something like that, to create domestic animals to do something for us, for our life. In line with the conference theme, I want to end my talk with a statement. Actually, the communication I emphasize today, communication is actually the life. We are not defined by our body. Our life, my life, is not as important as I thought. Actually, it's not important except for those impacts that I make to people around me. With that philosophical statement, I want you to enjoy the next 30-second video to remember this talk is about Cheshire cats. All right.
My head begins to jingle most every time I nod. 'Cause obviously, quite obviously, I was just a kitten. They said I'd be a gem, but now that I'm a Cheshire cat, it's odd how odd I am. Most cats have handsome whiskers, but me, I've got a
All right, that's all I prepared for today. I have 2 minutes saved for any questions you may have. I want to thank the organizer, my host, Regina, on the right side, and I want to make sure to know that scientists are having fun, and I want you to know that. My lifetime hero, Albert Einstein, said that. Thank you very much.